Perdón, se me fue para siempre.
Oh. Ahí le robó el
Ahora Pedro, porque probaste las slides de Pedro en sorry.
Okay.
So but you will allow them.
I will because I was changing last minute changes and that was very good.
Okay, so good morning and welcome to the third day of the school. So I think that people are becoming hard because we have reviewed the population at first time. So it's our pleasure to introduce Pedro Sánchez. So yeah, I guess that you're used to these kind of jobs, right? No. Okay, so Pedro did actually a couple of bachelor studies. Yes, the first one in chemistry at the University of Murcia, and he also studied biochemistry at the Universidad Complutense de Madrid. In 2013, he got his PhD in medicinal chemistry, again at the Universidad Complutense de Madrid, right? And then he did a couple of postdoctoral stays. The first one with Professor Federico Gago, who's in the audience, at the Universidad de Alcalá, and the second one with Leticia González at the University of Vienna. After this, in October 2020, he moved to the Department of Physiology, which is now called Department of Medicinal Chemistry at the Medical University of Graz, where he has a tenure track assistant professorship. And he leads the group of laboratory for computer-aided molecular design. His research interests are basically the design of polypeptides and protein with improved properties, especially to be used in therapeutics. And for this, he uses PLMM approaches. And I don't want to save you more time, so thank you very much for having accepted being a lecturer in the school, and yeah, enjoy.
Thank you very much for the kind introduction, Ines. I'm really happy being here in Madrid with this audience, with you and Federico. It's also emotional, this room. I mean, that was the room where, when I was at the physics, I mean, many of my colleagues, they came here for the PhD funds. There was really a lot of party around and also suffering. And I'm in Graz, in Austria, but originally I'm Spanish. Since 2020, I set up my laboratory in Graz. This is the second city in Austria. This is in the south, really beautiful, green. No one knows where it is, but I can say that it's worth it to visit. And today, I want to say also thank you to Ines and to Paco for bringing me here. I'm happy to share some notes about classical molecular dynamics. This is a technique we use extensively in our laboratory. Of course, we are in a medical university. We also do wet lab. We do applications. And to be honest, almost all the projects we are involved, we always put a little of ND or some kind of sampling. My question to you, just the first thing to say before going into detail is this. So if I show you this, what do you see? Anyone from Madrid?
strawberry tree.
Yeah, this is the symbol of Madrid. This is the bear and the
Strawberry tree.
strawberry tree. And this is, I'm happy that you recognize that, because this is exactly my model to tell you that is the also and Madonna, yeah. So this is basically one model. And the question I did to you, my model is working. It's really useful. But if I would ask you now, how is the temperature of the tree? How is the surface? How is it's really hard or not? My model will fail. And this is really like the leitmotif in computational and theoretical chemistry. So the question to the system will be the driving force to select the method we are using. And this is basically what I would like to introduce first about classical molecular dynamics. If we do just the representation, maybe you are familiar with that, but time scale, at the length scale, so we can say that classical molecular dynamics is to explain nanoscale. So we are talking between Armstrong and micrometer, and between picoseconds and millisecond in the best case. Of course, this is highly complementary with the sub-nanoscale using quantum mechanics, so we can really go small length and time scale. Basically, we use molecular dynamic simulations to study atomic models. Why atomic models? I will justify later, but in principle, you can say that animals for ND simulations are also big molecules like proteins and DNA, RNA interaction of these with a small molecule. Using these two methods in the sub-nano scale and in the nanoscale, we can go for the exploration of rare events like chemical reactions. Also unfolding and folding, that's maybe much more complicated with classical molecular dynamics. You need a lot of time to doing this and also to speed up the mechanism. And of course, there are complementary scales that we are not going to talk today about that. I think you have already cross-grained. I know this is coming tomorrow. Was it already there? And also macro scales. So of course, we can do many. Also, we know when we talk about classical mechanics that it's really cheap, it's really affordable, it's simple functions, and in comparison with semi-empirical methods, Hartree fault, EFG, post-Hartree fault with function methods, that all of them are much more expensive than. So, as I said, proteins, DNA, RNA are basically target molecules to use with molecular dynamics. And this is because biology is really complex, heterogeneous, and asymmetric. Here you see just one representation of David Butchell. This is from the PDB 101. It's really beautiful. It's just one illustration. This is not one structure. And this is one detail of one bacteria. So you can see a lot of colors, that everything is crowded, everything together, interacting, a lot of things. For example, you can see here like the DNA, you have some polymerases, you have also the ribosome, some proteins in light blue. So it's a scenario really complicated one. And also MD is really important when we have like a sum of molecules and interactions. Here you see like a representation of the GABA A receptor. This is in our brain in many places, and this is a receptor that has many binding sites. When we are developing a drug, we know that the drug binds to a molecule in a binding site. We talk about selectivity. And then there are some interactions, molecular interactions. And of course, these molecular interactions will end up at some point in the conformational changes in the receptor. Sorry, because the pointer is really one example of ligases. This is well known in biology, so that you have like one helix here on the top, one lead that it's going to move and to cover, and the active site based on the presence or the absence of some residues or some ligands that are a substrate that are there, or changes, for example, of the polarity of the solvent. So that means that to understand all these kind of interactions between small molecules and large molecules, we need chemical dynamics. Here you see the tables. Sorry for the table, I'm not a fan of table presentations. This is just an adaptation from McCammon, and I want to just to show here the different events that we are considering. Movement in proteins, we have the bond length vibrations, so some globular domains, how they vibrate, rotation of solvent exposed side chains, also torsional liberation of various groups, the groups that are not exposed to the solvent, and King's bending when I have like a loop connecting two parts, and this is just the part that can, like in a leaf, that can move, rotation of various side chains, allosteric transitions. This is really challenging, and also when you have some kind of unfolding, local unfolding. For all this movement, we have different recipes. I mean, of course, vibrations we know how to do with quantum mechanics. But for the three elements here, this is the adaptation of McCammon, we can use just plane and dissimulation. This is in the time scale, we can afford that. However, for these relevant motions, these really small, slow motions, plane and dissimulations are not sufficient. There are many problems. The first one is that this is a multi-minima problem. So maybe we are not able just to sample the whole configurational basis space. And also because I will need a lot of time, like millisecond time, and I don't know, but if the protein is really large, I mean, it will take a while. Usually in the 70s, and this is an idea I took from Pedro, he visited us in September in a workshop in Graz. Structures, I mean, people from X-ray, they're able also to see some kind of movement in the structure they have. And this is because they can just apply normal mode analysis to these macromolecules. And this is just a simple approach, a harmonic approach, where you just can expand the potential or the energy of the system, giving one structure that you are in a minimum. So you can set up as a reference state. So then the first term at that position, I can set up to 0. The first derivative is the minimum, should be 0. Then I can just take the second order term. then you have everything. So if I take now the equation of motion here, the kinetics and the potential energy component, and I apply just one oscillator function, then I can just end up in this equation where just the Hessian matrix here I have the eigenvector that are the normal modes, and here are the eigenvalues of this eigenvector that will be just the square of the frequency. So it just add up the movement to something that postulates. The amplitudes are not there, so I have to define the amplitudes. That's why when you compute normal modes analysis, you just multiply the vector. You can remove the same movement. You can do like so, or even you can deformate your structure highly. And here you see one example of the light base I saw before. There are two crystal structure, and I computed using just a lemon, that is the server, and from the from the open conformation, sorry. And you see that just with the normal mode 7, I was able just to catch up this movement almost. This method, in principle, it's expensive in terms, I mean, in comparison to what we would like to do really quickly. So let me justify. So if I do this, I will need to have like a force field. I will need to compute this energy, and this costs time. Let's say that. But of course, I can do just several approaches, like the elastic network model, where I can just reduce this potential energy just to this simple equation that is just not anymore like a single position of atoms, but just the pair. So just I define this B by J as the distance between pair. And now I can just compute the differences between the reference value, and that is much cheaper. And this is indeed how a lemma works. There are many ways known in structural biology. There are many servers you can use. I mean, like a first approach when you need to know the dynamics of a macromolecule. And I use a lemma, but now we are in Madrid. Maybe we can just highlight IMODs on the second slide. So what I mean with this is from a static structure, we can already predict some kind of dynamics. In the 70s of the last century was more or less the time where molecular dynamic simulations were like established as we know nowadays. And this is just one sentence I took from the first MD simulation that was reported from Acamon, Gellin, and Karpus, where it states that molecular dynamics can have a corresponding role for the internal motions of proteins. So they saw that it's possible to follow the movement. This is the movie. You can watch it on YouTube. Yes, of the first movie about the MD simulation of petroleum, involving pancreatic trypsin inhibitor. This is a really small drawing. It's around 58 residues, I think. And that was the first one where the hydrogen were treated explicitly. The simulation time was around 56 picoseconds. This is something that nowadays we do even in our laptop in a few minutes. So, in summary, more or less, molecular dynamic simulation is a technique that is used just to study complex and heterogeneous media at the atomic level. We can go really large, a million of atoms is not a problem. Sorry, I cannot move because the pointer is not. We can describe fast, moderate, and slow motions with a star, please, with a star there, not always the case. We can go to the millisecond time scale. This is now feasible with kind of architectures. And here, yes, I took like one figure from this review. This review is for experimentalists, nothing to do for the physicians. I mean, the journal is Journal of Neuro, I don't know, but really experimental. And as you see here, that the authors, yeah, they look for molecular dynamics as query on the title of papers on the top 250 journal. And as you see that the growth is really exponential. And why is this? What do you think? Why is suddenly this exponential growth?
Computational power. Computational power.
Yes.
We have more resources.
That's exactly that. So this is basically the contribution of the GPUs. This is one of the EPUs we have in our group. And if I remember well, I mean, the first implementation in AMBER was AMBER 11, maybe. So 2011, maybe. So I'm not sure, but it's more or less here. But of course, AMBER was not the first one. So then you can just see how the slope was. So in our group, we have many questions when we are using MD simulation. So for example, we want to know the flexibility of some parts of the coding. We want also to refine a structure in collaboration with our colleagues, the experimentalists. We want to know how is the binding of a small molecule to a macromolecule. We are in a medical university. We model drugs. And also, for example, Daniel is sitting here. One of the questions we are facing now is which residues are important for the activity of one enzyme. Can just anticipate that? Can we speed up protein engineering? So let's start just to do like an overview about the theory. I think I asked Juanjo that most of you are theoreticians, so hope it's not too boring. So these are just the equations, the Newton's law, equation of motion, is classical mechanics. So we can and also we have particles. So with these two equations, we can get the classical Hamilton equation of motions, where we can describe the time evolution of these particles. We will know the position and also the momenta at any time. However, these two equations, when we go to the multi-variable, really large system, it's really difficult to get an analytical solution. That's why we have to go for approaches. One first approach will be like the Euler approach, where we can just estimate or compute the position and the momentum at t plus delta t, and just took in just this extra term. However, this is really not feasible. It's a really, from the point of view of computational power, it's not feasible. We will discuss this later. But let me just talk about this delta t. This is the time step. And of course, this is a finite time step. And the question is, which is the value, the optimal value to that? So we have some kind of numerical stability, but also we can really go for large computational time. So imagine that we have this kind of space trajectory in black. This is the real one. Let's call the good one. And then we would like to approximate that just according to this equation. If we take a really small time steps, we can really just fit the plot. And more or less at the end of the day, we will have something that looks like more or less like the phase space trajectory. But as I said, this is quite expensive. But let's do now the opposite. Now let's take a really large time step. And if we do this, now we are going to end up in numerical stability. Because of course, if you see here the error in comparison with the trajectory and mimicking, or the trajectory I would like to reproduce, it's really large. So how small can I go with the time step? Well, there is already like the recipe, but at least has to be one order of magnitude smaller than the fastest movement or vibration in the system. And this is the vibration between a heavy atom and hydrogen, around 10 femtosecond. So in principle, the time step here should be like 1 femtosecond. We go for 2 femtoseconds, but I will say why we can do this later. In many of the codes that we have nowadays, I mean, the Euler approach I was so before that is not really efficient, is not used. Indeed, you have the velocity parallel, which is one of the algorithms used today nowadays. And this is just basically, I just connect the position and the velocity. I'm not talking anymore about momentum, but it's the same velocity. And it depends just basically on the previous position, velocity on the previous position, and the gradient. And the same for the velocity. There is a Berlet algorithm that was 1967, and in this case, it was also having inside the forward and reverse leapfrog algorithm. This is also used in diplomas. In this case, the main difference is that you have the position and velocity. They are like not in phase, like shifted, like 1/2 of delta t. And as you see here, for example, the velocity depends on this plus minus delta t, but the position depends on the t. So let's just simulate, I mean, a really simple way how everything works in the computer, like one possible scheme. In red, you have the terms that we know already, and in black, something that we don't know. So the first thing to do is just to compute the gradient. These are positions and these are velocities. So I can just define one delta change, and then I will compute the gradient. If I compute the gradient, automatically I will have also the position at the delta t. And then the next thing to do is just to compute the gradient at the new position because I'm already there, and then I will get the velocity. But this way I can repeat, iterate, and I will just integrate this for the whole trajectory. How I get initial conditions. And this is maybe one of the key points for theoretician. When you are playing with a small molecule, you can do really a couple cluster, you can really refine your structure, optimize, you can really get a really nice qualities. When you are using macromolecules, the story is totally different. You need to have a lot of knowledge about the biological system. And that means that you have to explore experimental models. The repository, the standard, is the protein data bank. You have different interfaces, the European, the American, the Japanese. For small molecules, also the Cambridge structural database. It's a really good starting point. Of course, you will need to optimize in the force field or using quantum mechanics. And of course, you can just go first principles. You can go theoretical models. You can draw somehow to get this structure. For the velocities, you can just assign, this is classical mechanics. So we have a distribution, Maxwell distribution. Depending on the temperature, you can assign randomly these velocities for the initial. I said before that in principle we could go till 1 femtosecond for the propagation. But there are some tricks there that you can go, you can increase the time step. And to do this, you can just remove degrees of freedom of the system. So it means that you are going just to constrain some kind of vibration, some kind of movements that are in the trajectory. And if you consider in a solvated system, like a protein, like a small molecule, almost 80% of the time it's because of the water of the solvent. So you can just impose one potential, which is the constraint potential, that will be unconstrained with something that is correcting the potential I want to modify. Okay, this is the constraint equation where I just set up one reference, one distance for a bond, and I just need to get this Lagrange multiplier for all the terms, so that I ensure that this sigma is 0. This is the expression that we have for the Pedro algorithm, for example. So doing this way, this is quite normal nowadays. That applying, for example, shake. This is one of the algorithms. You can go to 2 femtoseconds, for example. Another thing is now, if you couple your MMM with QMMM, you do QMMM, you will need to reduce the time step because of the. So we know already how to propagate. So the question, where is the energy from? I think everyone knows. Where is the energy from? In classical mechanics, it's a force field. Okay, I will do it. Yeah, it's a force field. And force fields are really simple mathematical functions that they connect the internal coordinates of the system with the potential energy. I like the definition that Hardy gives in his book. And first of all, when we talk about the force field, it's about these mathematical functions. So it's the equation to calculate the energy of a specific model system. So, fine. But also when we are talking about force field, we also refer, for example, the force field for protein, or a force field for sugar, or for lipids. So also when it's quite common nowadays, when we refer also to force field, it's like a set of parameters for these equations that has been optimized or fitted by someone. There are many force fields and software. Here, just this is not a complete screenshot, so just I put some of them. You have, for example, the assisted model building and energy refinement. So AMBA, we are going to play with AMBA today after lunch time. Hope you are there. I think it's the Spanish team is playing. Generalized under force field, also graph. Optimized potential for liquid simulations for OPLS, CHARM, GROMO. And we have also programs like Amber is a suite of programs, GROMAX, NAMD, Desmond, Schrödinger, and CHARM. This is just one screenshot from Federico, where you see the I would say the time evolution of all these phosphate that we know nowadays. For example, here you see, sorry for the color, you have here amber, you have here gromos, charm, and OPS from the others. As I said, everything started in the 70s of the last century. So, definition, phosphate is a simple energy function equation. We can, using phosphate, we can go to the atomic level. It's empirical. You have to calibrate, you have to fit the parameters, you have to compare with something. And usually it's either you take experimental data or you take also human calculation. So you need parameters. This is one of the questions for the phosphate where you have like two groups of parameters. The one is for bonded interactions and the other one for non-bonded. In the bonding terms, you are going to include the representation how you represent bonds, angles, dihedrals, other torsional angles, and non-bonding terms, you have van der Waals, hydrogen bond, Coulombic, etc. To do this, you have to just label your atoms, and this is what is called atom type. Atom type is the way how you can classify all the atoms, and based on your chemical intuition or chemical behavior. Let's play with the timing. We are going to use this molecule in the exercise today, and we can just start labeling them as the carbon. So here we can define, for example, the sp2 carbon on the carbonyl, the functional group as small c, the oxygen, small o. We can do the same for the aromatic carbons, cc and cd, also for the protons that are connected to them. Also for the NH groups like N, H, N, everything is small letter because this is gas. If we were using amber, phosphine, that will be like capital. But that means this is really well established. And one of the important principles of phosphate is transferability, so that they are compatible at some point. So in principle, I mean, amber and gas, of course, they are compatible, they are in number, and you can use them at the same time. Together with the atom type, we need point charges. And now this is where all the fun starts. Yeah, what is a point charge? Well, this is an artificial thing we do because we also already know how to compute electrostatic potential. Yeah? And then the question is where I cut in a molecule, in a bond, how I just define this scheme for the point charges. Here you see like this three play. I just wrote down three possibilities. Will you take 30, 50, or 60% of this electron density to A or to B? And depending on that, you're going to have a number. So which number is the best? How do you do it? Well, the answer is that there is no answer. So it depends on the phosphate. So if you're using, for example, amber, all the phosphate that you have there for amino acids, for sugar moieties, are all of them based on the rest scheme. If you're using OPS, all of them they are fitted to experimental macroscopic properties, charm, somehow reproducing the quantum mechanics interaction with T3 P water molecules, and in Gromov, it can be computed. That means it doesn't mean that you can, for example, use TFT to get the electrostatic potential because you think that it's better than Hartree Fock. But if later you're going to use a phosphate where everything is computed at the level of Hartree Fock, and now you compute the binding energies, you are including one artifact there. And let me just highlight or to show one example of us. During my stay in Vienna, we were interested in excited states. And of course, we were doing a lot of sampling, energy sampling in advance. Here you see one protein derivative. And then the point was to get the point charges on this cycle. Here I'm highlighting just the point charges for the oxygen atom. Just computed at the level, at the Hartree level as is in the amber phosphate. But then we said, okay, why not just use the GGA and DFT to make it. And then you see that the numbers, they change, not too much, but they change. And we can also go for just literally, everyone's using literally, why not? And we also put it there. So you see the variety of points of the point charges that we have. So be aware of that because this is important for because you are doing something. For example, if you are computing free energy. Bonded terms. Of course, we can model bond angles, and basically you go really simple. Use harmonic potential. Here for the bonds, and here I'm highlighting, for example, for this bond, that bond, you have to include so many parameters that you need as bonds in the system. The same for the angle. Sorry, I know you're theoreticians, but maybe some people on the internet not. Yeah, so here you see one harmonic potential. By the way, I like this GeoGebra. My wife is the teacher in the high school math, so she uses a lot of GeoGebra, and I found this interesting. So this is just one harmonic potential, and in this equation I defined the harmonic constant value of 10. So the question now, what happens if I go now to 1 or 200 for the constant? Yeah? If I go to 1, what would happen with the verb? What do you think? From 10 to 1. Are you following? Okay. So if I now draw the gray dot, that is very much broader. So this is how I represent like weaker bonds. If I have the value of 10 in this case, the oscillation there will be like very constricted to this shift in the coordinates. Yeah, this value here, I mean the gray one, the vibration, I mean the oscillation, they will cover a much broader range. And we can go also in the other directions, so we can really now even increase the constant. So now it's much smaller. But this is how force will represent, for example, bonds. We can assign the green potential for double bond, triple bond, and the light blue for single bond, for example. Now the question is how I come back. Okay, so. Yes. I have to hide this. Sorry for the inconvenience. No. So the main limitations of using these harmonic potentials is that they represent basically the real potential in around the equilibrium position, but of course, if we go really far away from this situation, the solution is they are failing. We can use also a representation for the dihedrals with an expression like that, where we have different parameters. A dihedral is just an angle between two planes. For example, I put this example there. We have the angle is omega, kt, and gamma. And let's see one example. For example, with one plane, and we can just see that n will be the number of minima that they have when I rotate. Then we have kt. This is the how large is this energy barrier. Here in kj, but in kcal is around 2.9. And gamma will just determine the angle. It's the angle that determines the position of the minima. We will check these terms later after lunch on the positive parameters we are going to do. Not bonding terms. So we know Coulomb, van der Waals, repulsion interactions between two atoms. We know that the Coulombic term is classically explained. And also van der Waals, so decays this way, and the repulsion decays that way. So if we put them together, so if I go fast here, so we end up in the Lennardian potential. This is something we were playing with from the first year of the bachelor. And you have just the combination of these two decays for the two terms. These two parameters, sigma and epsilon, here's basically the parameters I need to specify to get for the phosphate. So this is the expression, the bonded, not bonding. If I put everything together, I will end up in an expression like that, where these are the bond terms, where I talk about the bonds, angle, and behaviors. They are all the torsional angles, like for example, in propane. Imagine that you have one atom that is in a aromatic, one hydrogen, for example, from phenol. So if you want to define if this hydrogen is on the plane of the ring or up or down, you can do this with one propane. And the non-bonded interactions here, where you have this is vibrating but not moving. So the van der Waals, hydrogen bond terms, and Coulombic. Hydrogen bond terms are not explicitly there anymore. This is included in the van der Waals, in the potential, the linear term potentials, but there are some force fields where you have explicit term for that. For example, in the Rosetta binding Rosetta energy, this is a function that is used in protein engineering, for example. You can really play with a lot of terms, like for example, hydrogen bond. So we know how to propagate classically. We know how to get the energy from the force field, but there's still some aspects that are used in ND. So coming back to the initial photo and in the image of Futsal. So when we go to the computer, we just use a reduced system, so something small, but the reality is much more complex. So when we take these atoms away from our model, this cluster model, so we are going to find that in this case, in the representation of the whole, the percentage of atoms that are facing the vacuum, not too many, but if I take just this small system, the percentage of atoms that are now exposed to the border will be much higher. So you can understand now that maybe if we have 12% of the atoms that are now facing like a different interface, and I want to compute now properties like electrostatic, the properties there will be totally different than in the middle of the system. So that's why, but this is the main idea for periodic boundary conditions, just to try to avoid this kind of artifacts. We will see later, but for example, this term here, the Coulombic decay is really slow in comparison with the van der Waals term. And if this is just the border where I'm cutting up the system, you see that this term is not zero here. I have an error there. And this is an issue that we have to solve. So this is one animation of Juanjo that he left in Vienna. So what we do in periodic boundary conditions, we define the primary result, and basically you just multiply in all directions. And now what you do is when one molecule moves in this unit cell, then everything moves around. It doesn't make that you have different molecules, it's the same molecule. And the same if one of them is leaving or entering, leaving in this case, then you will automatically so this is the way how you avoid that the borders, the atoms that are in the border, they are not feeling like a different environment. But you are not computing the position for everything. You just need to compute the position for the unit cell. Of course, for computing the interaction, you have to check out one cutoff. And this is what I said before. I mean, when you are defining this cutoff, that you say that I'm going to compute explicitly the interaction terms for this atom here in this cutoff. And you see here how the two plots how decay with the distance or with the position. And you see whereas in the Coulombic it's almost zero. Sorry, I'm drawing that would be closer to zero. But if you compare with the blue plot, this is not the case. So we have like a problem with the Coulombic when we do this cutoff. For doing that, there are different solutions or approaches that have been developed during the years, like the evaluation and the particle mass eval. That is one version of this evaluation. Basically, the main idea of the summation is that I'm going to separate these interactions in a short range and in long range. This is the way how we can just compute the energy fast and also to remove this error, this decay, slow decay. The short range is calculated in a real space with the expression we saw already, that's the Coulombic equation, and the long range are computed in the reciprocal space. So imagine that now I can now define the potential energy like this short range and long range, and in these direct and reciprocal terms, and of course I need to add a third term to correct one artifact. I will explain why. In the real term, just I will just represent in this case just the point charges as we had before, and we have something new here. This thing new is just one distribution, one neutralizing distribution that is of charge but of opposite sign to the point charges. Because in the reciprocal term, I'm going to have this distribution, but in another time. And of course, this is itself energy term that yes, it's based on the point charge. Let me show graphically because with this pointer I'm somehow lost. So this is what we have. We have these point charges. We have 2 blue and 1 red. This is what I want to compute. So what I said is just going to the real term. I would add some artifacts now, something new, but this is neutralizing distribution I have exactly at the position where the point charges are. And now in the reciprocal space, I compute the same, but on another, a different sign. So when I sum up these two functions, I end up in that function. So this is the tree. So basically, I just I treat differently these long-term interactions. The particle mass eval is just on the top of the eval summation, but in this case, what we are going to do is just to use a grid-based chart distribution. So just as in many quantum mechanics methods, just using one grid, you can just put this point chart here. You can just create equi-spaced charges in a grid, and then you can go for a fast Fourier transform. That's much more efficient in the computer, and you can just really speed up the calculation of the long-term interaction. What time is it? Of course, when we are simulating, like biological system, we can have different phase or different situations. We can fix the number of particles, pressure, the volume, temperature, total energy, the chemical potential. But not all of these parameters or variables are independent. Here you see the different tastes that we have for simulations. You have the canonical ensemble where you fix the volume and the temperature. The micro canonical, that's really quite accurate to compute the thermodynamic properties. But in reality, what we are using when we simulate biological molecules is using NPT. So isothermal isobaric. So we have constant pressure in our lab, and this is how most of the simulation with proteins and DNA are done. So we need to control the temperature. We do this with one thermostat. And this thermostat is basically that you can define your system like a closed system with a particular N molecules and a total energy E is constant. And now you can just redistribute this energy or the temperature based on the Boltzmann distribution, so that the total energy is constant. So temperature will affect the kinetic component, and of course, by adjusting the velocities, we are controlling the temperature and the way around. So when we impose one thermostat, we want just to scale these velocities as I said before, controlling the velocities and controlling the total energy. And this is just by multiplying by this parameter, lambda. Sorry. And this is just irrestrained factor where I just can define in a simple way, for example, like a ratio of the temperature I want to target and the temperature that has my system at that point. This is a really simple definition, but there are much more efficient schemes. Like we are going to choose just the Brentsen thermostat and the Langevin thermostat. For the Brentsen thermostat, this term, this correction term, it's just a different expression where you have this tau. Tau is how the system is coupled with the bath that you have around, and depending on the value of this tau, we can have like a strong or a weak coupling to this bath. If tau is really, really, really large, then what we have is just looking at this expression, this term tends to 0, so we are going to have basically 1, so we have no coupling. So we are not re-scaling velocity. If tau is really, really small, we're going to have a really strong coupling with the path, and we are going to re-scale a lot. So usually for this time step of 1 femtosecond, the value of tau is around 0.4 picosecond, 0.1, 0.4 picosecond. All these parameters, when you're running this simulation, usually you don't care, they are default. But in some cases, you can touch and you need to touch. Another approach is going for a Langevin thermostat. Would you introduce friction into the system? So we are going to have now here these 2 extra terms. We have the friction coefficient because of viscosity and also random forces and conditions. So these parameters has been also specified in the fourth study. We can control the temperature, but also we can control the pressure of the system in the same way. I mean, we just re-scale the length of each of the sides of the box, and we can have expressions really similar as we did. I mean, this is the Brentsen thermostat that is basically following the same principle. So I think we are going to stop here. I don't know if you guys still alive. Because I know I saw I go really fast at that point. So let's make a stop of 5 minutes. And then we will see that. Next week.
Simulando como la que
So welcome back. After this short introduction into the basis of the theory of MD, let me just highlight what I consider the three main steps. I mean, from the point of view of operating main steps, we're running a simulation of a macromolecule. The first thing to do is just let's talk about protein, but you can put the DNA wherever you want. Let's start with the first thing is just to get the 3D structure of your macromolecule. The second thing, of course, the action or the important part is always the interaction with the small molecules. You might need to simulate the complex. And then later, how you simulate and you compute property, some average, some property on the top of the MD. When you go to this scheme, that's everyone will agree here. This is a common way, yeah. But you have a lot of points and questions to face. first thing is that we are working with biological molecules. I mean, so we need really, for the truth out there is that experimental models are really the truth. I mean, if you are working, I think many of you in computational theoretical chemistry, and if someone, for example, you are doing like a homology modeling, and then suddenly the X-ray structure is out there, and your model is, I don't know if you will be able to publish that. So experimental models are really important in the community when you are simulating biomolecules, but of course, these are models because they are biased by the operator. I will say later. I will talk about that. So that's why it's quite important for us just to curate and to iterate, to be really treat with love and care your model, and spend at least a couple of weeks like playing around, learning about your system, and try to see all this corners. Protonation. Protonation is also really important. This is one of the ways how biology is regulated, and of course, if this is regulated, this is because it will affect the conformation of this 3D structure. This is something we cannot forget in our simulation. How is the binding mode of the ligand? So this is really a question that you have to face at the beginning. How is that? Also, the protocol for DMD simulation, what I want to simulate. I'm working with a globular protein, I'm working with a membrane protein, working with DNA, the E4, so they are like in the quadruplex, that is the ending are really flexible. And then at some point my E4 will be like doing really weird things in the ending. Also, for how long I need to simulate, and how many simulations I have to do. There are many questions there. And of course, how I include the environment. This is maybe when I presented at the beginning the classical molecular dynamics. This is one of the key elements of MD that we can incorporate environment. We can go to really large. They are really useful databases when you're playing around with macromolecules. The first one is UniProt for proteins. This is like the reference, the database for proteins. When a protein is characterized, sequence is deposited there, and you have a lot of information around that. And importantly, in UniProt, these entries will be created at some point, so the information that you have there is really consistent. I highly recommend when working with proteins, for example, for ND, to visit UniProt. Brenda is a database that is much more oriented to enzymes, that you can have a lot of parameters related to catalysis of this enzyme. And this is, for example, really relevant for us when we are doing protein engineering. We said already, we talked about the PDB, this is the protein data bank. As I said, you have different interfaces, the European, American, Japanese. Here you have the first one is the American one, the second one is the European. PDB redo, it's a really nice initiative. As I said, these experimental models, they are biased by the operator, and of course, you have some issues that could be improved. And this is exactly the purpose of PDB. So already structures that are deposited is a PDB that be revisited by someone and improved. So it's you need when a structure, please go to PDB redo in a first approach. Also, with deep learning algorithm, this was on the news. It was like the revolution. We will know everything. Yeah, but be aware that this is just a model. Imagine, I don't know, like for example, one ion channel in a membrane that you have different open and closed states, and then the algorithm, which kind of state are you going to get from that? The open is closed and intermediate state. PDB sum, that's really nice if you want to navigate based on the secondary structure of the protein, like the topological elements. So imagine that you can enumerate all the alpha helices and also the beta sheets. So this is really quite nice when you want to explain and talk about your system, talking about a residue on the helix tree. So yes, in PDB sum you get really nice schemes, but everything is enumerated. PDB is a database, structural database, but the enzymes are grouped by activity, by family. So if you are working with serine hydrolases, or serine proteases, for example, you can look for the EC number of the enzyme, And then you get listed all the structure related or contained in this panel. This is quite nice when you are suggesting one reaction mechanism, because you can have like some of the other enzymes has been co-crystallized with one substrate analog, or maybe some kind of change at the active site. This is really useful. PISA server. When we get the structure from the, for example, from the X-ray, we have a crystallographic symmetry that is imposed by crystallography. But other thing is if our protein is a dimer, it's a tetramer in the real life in the cell. So that's why in the PISA server you can just check how is the molecular symmetry and escape out of this kind of artifact, because it's really difficult, challenging when you are just opening a primer sometimes to know if you have a dimer or you have a dimer of dimers, for example. So this is just the interface of the American paste of the PDB. Here you see like one identifier. This is for alphanumerical digits, and this is unique. So this is the identifier of one structure. Of course, one molecule can have many structures. You have different identifiers. This is new here. This is a Erlenmeyer task, and this is new in the PDB. That means this is one experimental model, because based on, due to AlphaFold, now all these theoretical models are also on the PDB. And what I wanted to highlight here is just these kind of plots from red to blue. To be really simple, you can think that red is really bad and blue is good. So, and then here you see some kind of statistics, some kind of parameters that our colleagues done X-ray, they fit the whole atom model into the electron density, and we can have a statistic about the quality of our structure. It's a really good practice to look not only at the whole atom structure, but hadrons, maybe they're not there, but also look at the electron density and how this whole atom model is fitted into this electron density. Experimental models. As I said before, we people in computational chemistry and theoretical chemistry, we have to face this every day. So if you get one structure release, experimental structure release, it's really difficult that you can just fight with your theoretical model. Here, this is one example when my time with Federico, where I want to show how our computational approaches help experimental people to solve this structure of one enzyme. Here, this is an alanine glutamase. This is one enzyme that is PLP dependent. This is just, you have here one L amino acid, then something happened. So you have like an internal, an external aldehyde, and at the end of the day, you have the D amino acid from L to D. So you are just changing the absolute configuration of the alpha carbon. So our colleagues, they found that when they got the structure, the PLP was degraded. And what happened when you see the arrow, the Fermi group was absent there. This group here was missing. At the same time, they also saw some kind of glow here, a mass close to the ring. And after doing some kind of suggestion of the reaction mechanism, different degradation pathways, we could assess and felt that this glow was corresponding to CO2, and that the reason of that, after also a lot of experiments, was that the effect of the buffer that we use with trees is a really buffer that people, when they experience a protein, they store the protein in that, was affecting and was indeed one reactant in the reaction. So you see here, the base is the pinky molecule there that was reacting and degrading the fuel. Protonation state. Nadia was really interested on computing the fluorescence of a dye inside the GABA A receptor. This is a really complicated scenario. So you have a lot of pharmacology there, but our goal was to try to understand how was the emission of this fluorescent derivative inside the binding site of GABA in the receptor. This molecule was designed by some experimentalists, and the idea was that coupling this dye with one antagonist. Antagonist means that goes to the side with the agonist, they are competitive, and it does nothing. So the effect is not inhibitor, it just goes there, so the agonist is not found. So they designed this for the specificity and neutrality, the binding site, and this for the dye. In the case of this dye, we had the possibility that this hydroxy group could be protonated or deprotonated. So then we did a lot of studies. We did also state dynamics, we did everything. But for the MD part, we just had this kind of solution. On the left, you see the deprotonated form. On the right, you see the protonated form. So the electrostatic there, I mean, the Coulombic was really important for the binding mode. And indeed, the 1A was binding much stronger. That makes sense. But what you see, just a single proton, in this case, just a single proton, same parameters, more or less, point charges, of course, not the same, but you see the solution is totally different. Residues in proteins. When you have a protein, you have some residues, as aspartic acid, glutamic acid, histidine, lysine, but they are treated very well with those. So that means the pH, I mean the protonation status of them, will change depending on the environment. And this is true, for example, for aspartic and glutamic acid when they are in a hydrophobic environment, really not exposed to the solvent. They usually are protonated. How we know this when we go for any simulation? There are standard protocols already. So you need to do this calculation. It's already you can use some servers like extract class, Dropcap, and then you will get the status of the protein for a particular pH. Dynamics and computing properties. This is the work of Alexandra. Alexandra is in Graz with us, with Daniel and me, and her project is based on trying to compute chemical shifts for small fragments bound through proteins. And all this is in the context of drug discovery. So that's why she's now implementing a workflow to do a much more efficient prediction of chemical shift using dynamics and human dynamics. So here you see the solution for the same molecule. On the top, you see the solution when you have just a single structure, that this is the X-ray solution, after minimizing and everything. And on the bottom, you have the solution when you take an ensemble of geometries, but in this case from human. It's not only, but human. What you see here is just, this is the error when you compare to the experimental value. Look now at the atoms, for example, 1, 2, and 3. And you compare here, when you take this ensemble, this dynamical behavior, the complex, the data is much better. Here I use just this non-covalent interaction of Julia, just to show you one movie. And what is important here, sorry for the quality you will see, there is a yellow arrow here, and this is highlighting or pointing to the hydrogen bond between a carbonyl group of the protein and one hydrogen of this imidazole. So hydrogen bonds are directional. This is not a perfect hydrogen bond, but in principle, this carbonyl group would be polarizing this hydrogen. But then we will say that in the context of the protein, this hydrogen bond is polarized, so it is shielded. But in reality, what we get is the other way around. From experiments, they see that this proton is shielded. So what is happening? So, and this is what I want to show here. You see in this oscillation, this is the carbonyl group. When we move the protein, this carbonyl group is never, ever close to this hydrogen anymore. And this blow that you see in green, this is the non-covalent interactions. You see here the scale, I mean how intense. And in this case, you see that now, so you came back to the beginning, but if we play again, you will see that this proton now is facing a lymphatic side chain. So now, at the beginning, we have one oxygen from the carbonyl group. At the end, we have just CH3 groups. And that's why we can explain now the effect they have in the experiment, but only after taking into account dynamics. And saying again that the solution we get from X-ray is not exactly the confirmation that they have in NMR. That's why dynamical studies like MD or ND are suitable and useful. Which is the role of the solvent in our MD simulation? So there is a really nice talk of Benedetta still on the website. It's organized by our colleague from the TU Graz. Some of us, we were together there. And I like the way that Benedetta classified which is the effect of solvents on molecules, that you can have like indirect effects. It's mainly affecting to the conformational phase space. Imagine that you have one residue that is positively charged, and then you have another one like a negative charge. If you simulate this in that phase, you will see that automatically impossible distributes will come together. But if you put some kind of solvent around some polar environment, maybe this polar, this positively charged residue will then expose it. And then you have also direct effect when you have like really direct interaction with your molecule, and then you have that polarization of the electronic charge distribution. You can do this. Basically, we have two models to go for that. We have implicit and explicit models. And here, just to say, at the did. First thing is, if we compare these two models, what is about the sampling of the environment, and what about the environment homogeneities and isotropy. So how our model represent that. For the first case, we see that implicit models are really very good to give an average of all the movement of the solvent molecule. Just one function, it's not explicit. And explicit, it's not so good because you will need a large time scale for simulating. You will need to go really large to see that this water molecule is able to move around. So you have at the end of the day one average for all the atoms. But then the other way around, if you have like hydrogen bond, the line that you have now, we will do later after lunch, we will compute how many water molecules are around some nucleobase. If we have like water molecule establishing one hydrogen bond with one of our hydrogens or oxygens, then in an implicit model, we don't have this water molecule. Indeed, there are some approaches that are like hybrid, where you have just a single water molecule, few of them in an implicit solvation model. But of course, so that's what they say. That's the job for explicit solvation models. You can choose many water models. For example, this is one snapshot from the PDF of AMBA, the manual. I think this is AMBA 18 or 19. And you see here how many different water molecules you have out there. Of course, you have also models for different solvents, and if not, you can parameterize your own solvent and you can simulate it. Water molecules are really special because they have a large dielectric component constant. They are really important in the biological context. And also, we know all about these hydrogen bonds. When we model water models, we have to get the number of charges, it varies, how many I put, how I get the interactions out of the dipole moment. Here you have the summary for 3 water molecule models, SPC, TIC3P, and TIC4P, and the different characteristics. For example, here you see the value for the angle between the 3 atoms. You have also the point charges. So you have that. Here, for example, you have the dummy atom, the P4P. But all of them, they are fitted to explain things. And what they explain, you compare with some kind of experimental or observable. So like, for example, specific heat, set, viscosity, or thermal conductivity. And you see the error for the specific heat is not so large, but for the other, the error is really large. For AMBA, the new force field that is 19 SP, if you go to the manual, there is a recommendation that please use OPC model. And this is because they also consider that this way of representing the molecule here has a 3 charts, 4 points OPC water model. So you have Q, Z1, Z2, and Y to define as parameters. They explain much better, or they are really better solution to use together with the new force field as in the current ones. here in this plot, you see different things, how different water molecules. You will have the slide, so it's really small, but basically this is OPC. You see the bar, it's almost not existing. And the other case, the error is much larger. Of course, it's their own paper. They show that it's superior, but let's believe them. We can also model liquids. I mean, it's not the same as we are playing with a globular protein that has transmembrane protein, everything changes. And there have been in the last year really a lot of postulates around lipids. This is, for example, the lipid 21. This is in amber. You have the different lipids that you can have in the membrane. But I highly recommend the membrane builder, or CHAM3, that is really compatible with amber. And it's really straightforward. The only problem there for you will be just to define the composition of the membrane, also to reset the center of the protein. You have like OPM, I think it's the server, because for specifically for the workload you have to do that. But after that, you will get the topology and the coordinate files for your simulation. So everything ready to go. Let me talk about just how it works like MD simulation. So there is usually the step that you have is the setting up of the system. We talk a little about that. This is basically based on the scientific question you want to deal with. You have to choose, I mean, which is your solute, utility, for example, which kind of solvent you want to use. And of course, you are going to use counter ion. If you are using potassium ion-gated channels, of course, you will need to take care about the concentration of potassium chloride, for example. The next step is about minimization of the system. So this is just basically you relax the system. You are just putting things together. And you are using your simple function, just no crash, for example. But of course, you have a lot of tension there and really positive energetical terms that you need to remove. And to do that, you will need to impose some kind of constraint. You cannot just minimize everything at once. I mean, of course you can, but the problem is usually this space. So I will talk later about that. Heating, this is how you start to put in some velocities there. This distribution, we saw at the initial velocities, we can just follow this Boltzmann distribution. And of course, for that, you will need also to constrain your system. Equilibration, this is just how you start imposing the conditions you are going to use in the production phase that is coming later. And of course, you are going to change from the example that you apply. Production is the main part of the MD simulation. So you have to be sure that you are computing or measuring properties in one region of the MD simulation that is stable under the condition of simulation. And the numbers that you are getting are representative. The number, you will get always number. And finally, the analysis. Yeah, about minimization is not getting the global minimum. We are not playing with small molecules anymore. We have a really big system, and as I said, we want to just remove some tension from the system. And for that, usually, in most of the MD software, you use two algorithms. The first one is the steepest descent. Sorry, the S is missing. And the second one is conjugate gradient. The first one is just following the gradient. And you just take one direction, you go for the gradient, and since the first derivative is 0 in the minimum, you just follow this gradient till you get the value of 0. And then now you take a perpendicular direction and you go in another direction. This is what you see is green arrows, always perpendicular. The problem of that is that you need to take many steps. So it's really slow. I mean, when you are at the beginning, it's really good to escape from a bad situation at the beginning, but later to get the local minimum, to find it, it takes a lot of steps in between. Indeed, sometimes because of this perpendicular nature, you lose a lot of, let's say, optimization there. And you were already in a point that were lower in energy, but then you end up in a situation that you have to come back somehow. The other one is the conjugate gradient that is recommended after some cycles of the steepest descent at the beginning. And it's much more efficient. As you see this red vector, the second one goes directly to the minimum really fast. Please visit this YouTube video. I think it is really good. It's really consistent in terms of mathematics and also really well, you can follow really easily. Here, this is just how you decide these steps. This is just the position, the new position that you are going to move the electron. I mean, from here to here, this is the first one. And that's the second one. Sorry, this is the first one, and this is the second one. And this is basically the module of your vector, how far you go. And what the algorithm does is imagine that you follow the negative gradient, and then suddenly you end up in a situation where the energy, I mean, the gradient is larger in terms of absolute value. Then the next ones will be maybe smaller, like in a previous direction. I put some parameters. We are going to discuss this later. First too much time. We will see later after that. We will play around this. What is the meaning of that? Heating. This is just like increasing the energy of the system by scaling the velocities. And for that, we have different thermostats, as you saw. And some practice, they start heating up from 0 Kelvin to 300 Kelvin. 0 Kelvin is a really unrealistic situation. Imagine that, having a probe at 0 Kelvin, I guess the X-ray structure was not obtained at 0 Kelvin. So it's a good practice also to start around 100 Kelvin, it's not a problem. And it's also a really good practice to have some rounds. So you gradually increase the temperature in a linear fashion. Because if not, you will see in the algorithm, if you define from 100 to 300, just in the first step, you will get the 300. And the rest of the heating phase will be just oscillating around. Equilibration, as I said, this is the way how you just end up in the conditions for your production phase. And usually the heating, I forgot to say, you do at the temperature and at constant volume you're heating up. This is the way you can just, later, in the equilibration, you can switch from constant volume to constant pressure. And if you do this, if you change to constant pressure now, you will see how the density of the system will increase. And that's good because if you're simulating water, you will see that slowly you go from 0.6, 0.7 to 1. That's what we expect for the density of water. And finally, the production. The production is just, as I said, you can have many situations, you can constrain, you can accelerate your MD, you can use whatever you want to do. The question, for how long should I run one MD simulation? That's the question to you. For how long should I run an MD simulation?
That's the case of substitutes.
It depends what you are looking for, but for how long?
Depend on the system. Depend on the size of the system.
Depends on the size of the system.
How many GPUs do you?
How many GPUs that everything?
The energy is aligned.
Yeah, that's usually happens really easily at the beginning. You measure, not only energy, but also, for example, geometry by RMSD. And usually in the first nanoseconds of the simulation, you are already there. But the question is, there was a tricky question. It depends on the system, it depends on the number of GPUs. So it depends on the capacities that you have. It's not the same that you have CPU-based core, and you have like an ion channel with memory. You will get 1 nanosecond per day in your computer. But if you have a GPU or a quantum machine, of course, you can go from the second. But the main question is the scientific question. If I am looking for a conformational change and it's not happening, I'm simulating one ion channel, I don't know for how long, but I see nothing, not yet. So for me, it's clear that the MD simulation is not finished, or maybe I'm just wasting my time doing that. So I'm not sure still about the answer. That maybe is the second one. But important is just what is better, to have a single trajectory, really long one, or just to have different independent simulations, but really this time. What do you think? You need more, sorry?
More than one trajectory.
Yes. That means you can have independent simulations, and at the end you treat the data as a whole, like a same conformation of the space, because this is reality. And this is the problem of MD simulation. It's highly deterministic, and you don't know if you can escape from one minimum if you're doing plain MD. It's a really good practice to randomize, for example, the velocities at the beginning, or to start from different initial conditions, run trajectories, and later analyze all of them like a whole. You can do clustering, you can do whatever. And this is, in my opinion, always a good practice to run 3, 5, 7 independent simulations. But of course, depends on the capacity, how much memory you also have. The analysis, there are many tools out there. So you can really, you can write your own script, everything, but all the MD simulation packages, they come with this kind of software. MD-Trust we use a lot in the lab. CPP-Trust, this is number. High-Trust is Python version that you can use with the Jupyter notebook. It's up to you. You decide what you can use. Perspective about MD simulations. So, okay, this is a classical method, but is there something new that people are now fancy things in MD? Yeah, there are many cases, many examples. Don't kill me, I'm not going to cover all of them. I will just highlight some of them. And the first thing is we come back to the table, the ugly table at the beginning. We saw that some of the slow movement that require like really low and large simulation times. But maybe I can do a trick. Maybe I can put some kind of elements or ingredients in my MD simulation, and I can try to speed up the process of sampling. These are known in-hand sampling MD methods. One of the ways I can do this is just by modifying the potential energy surface I'm exploring. And if I do this, I can classify, this is like artificial classification, like methods there where I know where I want to go, I know the endpoint, and methods where I don't know where I want to go. Yes? In the first group of methods, I am the one defining the collective variable. And I can use a simple, so simple definitions as distances, a linear combination of distances, one angle, RMSD, so it's a or patterning of a sugar. There are many different approaches. One of them is metadynamics, speed, molecular dynamics, simulations, umbrella sampling. And there are other group of methods where I really don't know where I go. Here I just selected metadynamics and replication, molecular dynamics. For the first of them, this is we did recently one study using metadynamics. In this case, it was QMMMD, but the principle is the same. And this is basically the potential that you're imposing depends on the previous one, and you have this term here. This is just repulsive by the synchronous. These are Gaussian functions that are controlled by this function that is here. That is, you have to parameter that is gamma. This gamma parameter is positive, larger by 1. And depending on the value of this gamma, I can have the situation when gamma tends to 1. Then this term, this term goes. And then I have basically, I'm not imposing anything to the system. So I'm not changing this energy potential. If gamma goes really large value, then I will have I will be in a situation to mind that gamma tends to infinite, and now I'm going to energies that are here. So I'm exploring in this barrier. So I find no energy barrier anymore. I'm just flying. So then I'm not sampling the energy barriers. I took here just the movie from Wikipedia. I don't like Wikipedia, but I like the movie. And here you see just how it works, this metadynamics from Pedro. And this is basically just as adding sand to the potential energy surface. Here you're going to see that we are starting its minimum on the left, then we start to increase via Gaussian functions. And the good thing is that when I'm adding Gaussian function, the next one will depend on the previous one. So I can change the shape, I can change the amount. And in this, sorry, I was talking. So then at some point, the energy, I can jump to the other minimum. So at the end of the day, I'm exploring all the minimum. Here, this is just counting how often I'm in one of the positions of the dynamics. I know how to close this. So This is one of the examples we are using now. So we are at a medical university, and we have interest in different projects. One of them is about alpha acidosis. This is a rare disease. It's a lysosomal storage disorder. This is caused because one of the enzymes in the lysosome, all the serial killers are there in our cells. They are responsible of degrading the things that are not working, and they are sent to the lysosome. So this is one glycosidase. This is just degrading monoacids from glycoprotein, and when this enzyme is defective, the activity, then we have accumulation of these oligosaccharides, and these end up for some particular mutations in a really difficult situation with severe consequences and really short life expectancy. There is no permanent cure. I mean, there is not a final, real cure. So you can go to the hospital every week to get recombinant enzyme. So they just inject, and you are there for 2 hours every week. And you can also go for bone marrow transplantation with all the risks that is involved. So that's why we thought maybe we could just try a different approach. Maybe we can just try to go for looking for molecules that are able to bind allosterically to the enzyme and to reactivate the activity on defective enzymes. We don't know if we will be successful, to be honest. But the point is, imagine that we can reactivate defective enzymes in vivo. That will be the situation of going to the hospital every week for 2 hours to get the enzyme that you just take it with. Of course, we are not going to develop the field, but this is the really ultimate goal. And this is what Bruno is doing in the laboratory. The first thing we needed to do is just to try to understand the reaction mechanism of the enzyme, and for that we use metadynamics. It was not just plain ND metadynamics, but PMMM ND metadynamics. So with that, we could characterize the enzyme transition state complex. We could just explore and get the energy of the process. That is at the DFT3 level of theory. We know that the energies are not really good. Geometry is not so bad, but the energies are not so good, and we are now exploring much more expensive methods. We deposit the paper and we can archive if you are interested. Another kind of technique we are using a lot in the laboratory is stable molecular dynamics. This is just, you just take one atom and you force the atom to go from A to B, and by applying one harmonic force in this case. So this is the energy term that you want that you have coming in this case, for example, from the force field, but then you have one extra term that has this harmonic expression. And this method is highly deterministic from the initial conditions. And then, of course, for example, for the reaction mechanism, you can have these energy barriers, and you have a huge variety of energy maxima, but this is because of the initial condition. If you minimize at the beginning, you can reduce this noise. And also accelerating molecular dynamics. This is one of the methods, as I said, that we don't know where we go. And in this case, what we are going to do is just basically just to define a threshold of energy. So we are going to put upstairs the system. And we have two situations. If we are below or over this threshold. If we are above this threshold, it's on the system that says, okay, no extra energy term is needed. If we are below, then we will put some extra terms so that the system can explore region of high energy. Of course, there are many applications of ND. This is really a unique technique, and nowadays it's combined with many different types of computational methods. For example, we can just define like a list of residues that they can have different protonation states in our dynamics. We can define like a multi-topology file. We have for each of the histamine, for example, I have the different definitions depending on the protonation state. And then I can somehow define the total pH of the simulation. And these residues can adapt so the total pH is given. This is this constant pH in this point. We can also just plug our ND code with the PMMM scheme. Usually we are using AMBA because it was really well developed. We have a lot of interfaces, many external programs. With GROMAX was at some point was not anymore there from after GROMAX 5, but now they are back. So this is just one screenshot this morning from the main website. I have, to be honest, not experienced because we focus number, but I think it's worth it to be explored. Also, free energy factorization. We can just use chemistry, chemical methods, for example, for the binding of drugs to enzymes or for protein evolution, which is the impact of single residues, and everything is classical. Finally, fossils are not thought to model reaction mechanism, but of course there are reactive fossils out there. The problem as well will be the parameterization. How good is your parameterization? And maybe React's effect is one of the clear examples. It's also already can be used in AMBA. But also you can use forestry, for example, for excise states. You can just also you have like a ball of equivalent and S1, so you can optimize on the minimum of the S1, the first excise state, and then from there you can get the point charges for your parameters. Yeah. Sorry, I was mistaken with the audience. Usually I face a more experimental audience, and that's why I took most of the things. And the bibliography I put here is pretty basic, but it's really well written. I highly recommend the book of Harvey. It's maybe not more for science. It's really well written, and I like the book of Kramer. It's a really nice book, and I use it for the teaching. I consider it's a really nice. I put a lot of references there. You will have the PDF after the talk. And yeah, I will finish as I started. So everything we do in the computer are just models. I think they're really useful, but also they're at the same time really dangerous. So it's a question of not forgetting that they are just simple models. And also, many of my colleagues, they're maybe doing like upper cluster things, really expensive things. Usually the classical part is like what? It is like really inaccurate. But I think they're mistaken because yeah, less accurate, but of course you can get different answer. And indeed, it's really amazing now seeing like a pure human people moving now in the direction of using sampling with classical mechanics, and looking now at people from classical world moving using, for example, PM with human skin. I think the future will be a combination of many tools and techniques, and I think that MD will be always that. With that, I would just to say that after lunch, this is enough and a lot of sugar. We're going to have like exercise. I'm planning. First of all, we are going to parameterize one molecule for a fossil. We are going to use the state like that. After that, we are going to simulate in a box of water molecules. I will show you how to build up one system in AMBA. And then finally, we are going to compare the situation of this ligand. This is a nucleolase in the with the biological context. We are going to take a double-stranded DNA, 12 of the strains, and we are going to simulate in a box of water. And then we are going to look at, for example, how is the water shell around the nucleolase. We're going to look at the RMST. We're going to look at distances. We're going to discuss a lot about the biology and the structure of the models. And with that, I'm done. I think we have still 12 minutes. And so thank you very much, and I will be happy to answer questions.
Okay, so it's time for the so regarding the metadynamics simulations, for the gamma parameter, I've read that for classical biological it should be, or maybe bias factor should be around 8. Then, but since you're doing also human part, is it the rule of thumb to kind of set the parameter without? Yeah.
I mean, we do a lot of trial and error. I like to be mistaken, like to learn. And to be honest, in metadynamics, we just started from something much more advanced. So we were the co-author of the paper is expecting metadynamics. So all these parameters were from the beginning. Metadynamics for human, but they were already there. So we were not discussing this point, but of course.
I have a question. So you said that you prefer to run several independent simulations instead of one long one. Indeed, I prefer the other way around. Because in this is equivalent, right? Because if you run a very long simulation and you split it in short fragments, it's like independent simulations. Because if the simulation is long, the memory is lost, right? At the end, it's the same approach.
I mean, if you go really long time, of course, it's as you are like losing what you are very at the beginning. But I don't know, my experience is I get much broader exploration when I go for independent simulation. But of course, each of them maybe in the microsecond aspect. So really large.
You have more GPUs than we have. Okay, cool. Okay, this is not the
See
case.
you later.
Yeah,
I
we
think the
can.
match is after my Spain is playing today, so I think it's after the drinking side. I hope I will see you.
Sí.